Hi everyone. Today I'm going to talk to you about the neuronal changes associated with aging. So, first, I wanted to point out something really important about neuronal changes. Neuronal changes are selective and subtle. So, basically, what that means is that they only occur in specific regions of the brain, namely the PFC, your prefrontal cortex, and your hippocampus. Hippocampus. And because they are so selective and the changes are so subtle, even though they're just selective and subtle, the structural and cognitive changes that are associated with them, or actually, I shouldn't say associated with them, but these changes, the selective and subtle neuronal changes, have a far greater impact than all the structural and cognitive changes that take place in aging. So the PFC controls your executive functions and your hippocampus controls encoding of memory. That's really important because changes in these places specifically have such a great impact on your brain, as you can imagine. So let's get more in detail into what changes actually occur. So in order to do that, we have to take a deeper look at what we thought were neurons and more specifically dendrites. So dendrites, we just thought were okay, if this is, I'm going to drop here, if this is your cell body, this is your axon terminals, um, these branches are your dendrites. And yes, there are many, many more, and they're very branched. They have multiple breaks. That is very much correct, right? This is what a basic description of a neuron looks like. But if you take a zoom in, so let's take a zoom in to one of these dendritic branches, just one. Let's take a look or zoom in to something like that. Well, if you zoom in, you're going to see many, many more branches in that branch separately. So if you were to kind of visualize this, let me just make sure you guys are understanding because this is very important. If this is your dendrite, this red outline is your dendrite, these little bumps on the surface of a dendrite are known as dendritic spines. So I'm going to just stop outlining because I think you guys get the point. But these little bumps over here are what are known as dendritic spines. So let's define what a dendritic spine is. A dendritic spine, okay, let's just do it here. A dendritic spine is a protuberance, from a dendrite, which reaches out, or I'll just write underneath that expands and expand and then retracts to form synapses. So you're probably a little confused right now because what do you mean a dendritic spine forms a synapse? Well, when we've said that dendrites form synapses, we haven't been fully accurate. It is not a dendrite that forms a synapse. It is the dendritic spine which connects with the axon terminal to form that synapse. You can kind of see here how this dendritic spine forms a synapse with this synaptic terminal. So why is this so important, right? Well, first, before I get into that, I just wanted to show you guys a really cool imaging. So this is like a dendrite under a microscope, and you can kind of see how these look, I like in real life. This is just a drawing, and this is what it looks like in real life. Well, your so back into why dendritic spines are important. Dendritic spines are one of the reasons or most closely associated with our idea of learning, right? So, in order for us to form connections, These dendritic spines act as microphones for signals to try and look for these new axon terminals. They'll literally reach out, branch out, and if they don't find anything, they'll come back. But if they find something, they'll form a synapse, and that's how we learn, right? So these dendritic spines are basically little microphones. Now, that's really important, right? And we'll get into that. You'll see why in just a bit. So I'm just going to write microphones down here so you guys can remember. But now that we know what a dendritic spine is, I just like to think of them as like assistants of the dendrite, if that helps you think about it a little bit better. To complicate things further, there are different classes of dendritic spines. So I want you guys to think of structure versus function, right? I know you guys have probably heard this a lot in AP biology, but structure is very commonly associated with function. And the reason for this is I'm sure you guys can think about it. If a goal of a dendritic spine is to extend and retract as fast as possible, the shape of the dendritic spine is very important to that, right? And therefore, I want you guys to think really quickly. Which of these do you think would be the best at extending and retracting? If you guess the thin spine, you would be correct. So, okay, I'm sorry, fastest. And which one would be the slowest? Actually, yeah, you're right. If you guessed stubby, you would be right. But for the sake of this video, we're just going to focus on mushroom spines. So basically, thin spines and mushroom spines. These are the two very important ones because when scientists conducted a study on aging in rhesus monkeys, you're going to hear the word rhesus monkeys a lot in any type of neurological studies because their brains are pretty similar to humans and they're just a very good model of what human brains are like. But in rhesus monkeys, they found that these two were highly affected by aging, more specifically thin spines, right? So they found that thin spines became less complex or they were less amount of thin spines. And let's talk about why that's so important. Well, given context clues that thin spines retract and expand and retract fastest, what do you guys think the function of these spines might be? Well, if you guessed that it was probably implicated in learning or more specifically working memory, you would be correct. It is most commonly associated with working memory because of how quickly it expands and retracts. It's also associated somewhat with learning, and it's really its ability to expand and retract so fast is just crucial to the brain, right? But what tends to happen as you age is that your neurons or your dendrites tend to lose complexity. So when we talk about complexity, all we're saying is just it tends to lose the variety of these dendritic spines. Using the variety of dendritic spines basically stops how much it slows down your processing power, right? Because it's not able to make the connections and retract as fast as possible. And it's just your working memory ability goes down. Remember, we talked about that in cognitive changes, and that the reduction of these spines has been proven to be really important to this whole theory of aging. So what you need to take away from this video is that there is one specific class of spines known as thin spines, which expand and retract very fast. And because of their ability to expand and retract so fast, they are very commonly implicated with working memory because of that temporary connection forming and separation, right? That's what we know. That's why we can't store like information for that long when it's in working memory. As you grow older, the rhesus monkey study found that your neurons lose complexity, and more specifically, your thin spines decrease, right? The amount of thin spines decrease and they become inelastic. They can't expand and retract as quickly. So I'll say that they become inelastic. And because they become inelastic, they're it's basically stopping your learning, your processing power, so on and so forth. Your mushroom spines are also affected, but these are slower contractions. That's really what you need to know. One thing that I wanted to add at the end of this video is a little bit about neurogenesis, right? Neurogenesis. So, neurogenesis basically refers to the creation of new neurons, creation of new neurons. The thing with neurogenesis, though, is that as we grow older, the rate of neurogenesis obviously tends to decline. Uh, tends to decline. Um, Keep in mind that there hasn't been any studies directly correlated to the decline of neurogenesis with cognition, but what they have found is that a decrease all these cognitive skills or cognitive enhancing activities tend to increase rates of neurogenesis. So although the decrease of neurogenesis isn't directly correlated to the decrease in cognition, enhancing cognition tends to result in increased neurogenesis. Make sure you have that order, right? Just as a quick review, there are only two places in the brain which continue to have neurogenesis into adulthood. The first one is your olfactory bulbs, and the second one is the dentate gyrus, which is basically just your hippocampus. Okay, so I hope this video was helpful. Please make sure that you watch all previous videos if you need to, and make sure that you're constantly internalizing this, constantly reviewing it, and taking away the key points. These are meant to be a supplement to the brain facts, so please make sure that you're also taking notes on the brain facts book too. Thank you. Bye, everyone.